There might be a little bit different version of them because I've been working on them before the lecture and I might not have uploaded the final version, but they should be roughly the same. So we're going to be talking about how cells regulate gene expression. So we have all this DNA in our cells, and we're not constantly making all of the protein encoded in our DNA because that'd be very wasteful and it wouldn't allow cells to specialize, because we know that all of our cells have the same DNA, but some of our cells do very different things. Like we have cells in our eyes that change color over time, apparently, and we have cells in our muscles that are contracting to allow us to do things, and our heart that's always beating. So we have all these different cell types that all have the same DNA. And the way that the body does this is because it regulates how our genes get expressed in different times. So we're going to talk first about prokaryotic gene expression. So again, prokaryotic cells are simpler. So if we talk about gene expression and how it's regulated there, it's a little easier to understand, and we're going to extend that to eukaryotic gene expression regulation. So we're mainly going to focus on two types of prokaryotic gene expression, what's called inducible and what's called repressible operons. And so we'll get a little more into what an operon is and what it means for something to be inducible versus repressible. So again, regulation of gene expression. So a gene is expressed when the information in the DNA is translated into a protein. So this process of going from DNA to RNA is called transcription. We make an RNA transcript of the DNA, and we process that by adding, a poly-A tail on the end, a 5' cap in the front, and we chop out different regions, chop out the introns and leave the exons. And that final copy is then sent to a ribosome, which attaches to the mRNA and creates a protein. And that whole process is gene expression, because expression is when we go from the information in the DNA to like the functional unit, like the protein. Because the protein can really be changing the cellular environment. It can really be acting on what's around the cell, and it can be really like doing different actions. And so the gene expression is like going from information to actions. And so the cell only wants to be making proteins when it really needs to be doing that action. Otherwise, it doesn't want to waste the energy and resources, and it doesn't want to mess up all of its kind of complex behavior. So we've talked a lot about enzymes, and so enzymes play a role in like all of the processes that we've been talking about. So photosynthesis and translation, enzymes are necessary to synthesize the biomolecules that are instrumental in cellular respiration and also in translation. So without those enzymes, cellular respiration and translation, they couldn't occur. So if we didn't have the enzymes to transcribe our DNA and to translate it into proteins, we couldn't make that happen. If we didn't have, sorry, I said cellular respiration and translation. I meant to say photosynthesis and translation, but it's also cellular respiration. In photosynthesis, we have a lot of enzymes that are involved in building up that glucose molecule from the carbon dioxide that the plants inhale or absorb from the atmosphere. And without those enzymes, we couldn't create that glucose at the end as our final product. During cellular respiration, enzymes are necessary to break down that glucose into ATP. So one enzyme in particular, we have that ATP synthase. So that's involved in creating the ATP from the proton gradient that we generate during the electron transport chain. So that enzyme has to be produced by the cell before it can undergo cellular respiration. So these enzymes are necessary to make the cell do certain actions. During mitosis and meiosis, enzymes are instrumental in these processes as well, and so they help the cell grow and they help it reproduce. So enzymes are types of proteins, and these proteins are created from information that's stored in the DNA. So when these proteins are expressed, when these genes are expressed and these proteins are made, the cell can do all of this different behavior. And how many of these proteins are made and when they're made influences how all of these behaviors happen.
So technically, is the RNA like an exact copy of that DNA segment that it's looking to express?
Well, it's got RNA nucleotides, not DNA nucleotides, and also it gets processed by cutting out the introns and leaving the exons, so it's got some minor changes, but it is a copy of the DNA.
Okay. I was just curious, because it's like almost as if that section of the DNA could function without the rest of the DNA, but it's kind of different because it's the RNA.
Yeah, so I mean, the RNA gets constantly degraded, whereas the DNA doesn't. So we'll talk more about this towards the end of the chapter, but this is another way that the cell can regulate gene expression. So does everyone kind of see the importance of gene expression? Everyone excited to learn more?
Yes.
That was resounding, yes. So again, we're going to study it first in prokaryotic cells because it's a little easier for us to understand it there, and then we can extrapolate what we learned there into eukaryotic cells. So bacteria have to regulate their gene expression based on what's in their environment. So bacteria can't really go to the grocery store and buy whatever foods they need, so they have to just sense what's in their environment, and they have to act accordingly. So if they see a biomolecule in their environment, they don't have to go through the trouble to produce that on their own. They can just take it in from the environment. If there's something they really need, like glucose, from their environment that they don't see, then they have to find another way to consume an energy source that they can use to make ATP to do lots of other useful things. So bacterial cells sense what's in their environment, and they act accordingly in a way to conserve resources and be as efficient as possible. So the operon model of gene regulation is a model that discusses some components of the DNA and how other molecules can interact with the DNA. So to start with, we have the promoter. This is the region where RNA polymerase is going to attach to the DNA to generate an RNA transcript. So what do you think would happen if the RNA polymerase cannot bind to the promoter? Do you think that gene will be expressed?
No.
No. So if we can't make an RNA transcript of our DNA, we can't express that gene because nothing can go to the ribosome to make the proteins that we need. So the way that we can regulate gene expression is that we can either allow or not allow by blocking the promoter with another molecule. And that molecule is called the repressor. So the repressor can bind to our promoter and block it. And if it blocks the promoter, then we can't express our structural genes. So the structural genes are the genes that do the action that we're interested in. So before I was talking about, bacterial cells sense what's in the environment. If they don't see any glucose there, they're going to have to make some glucose. And one of the ways that they can make glucose is by making an enzyme that breaks down lactose, another type of carbohydrate that we'll get more into later. But the enzymes to break down lactose are shown here in ZYA. I don't know why. It shows XYZ, but I guess ZYA makes sense as well. In ZYA. And so those are our structural genes. Those produce enzymes that can break down lactose, and so they're really doing the action that we're interested in. And so those structural genes are kind of under the control of the promoter. If our RNA polymerase can't bind to the promoter, we can't express our structural genes. And the RNA polymerase can be blocked from binding to the promoter by the repressor. The repressor is a molecule that can bind to our promoter, and it can block RNA polymerase from transcribing. Oh wait, I mixed this up. Sorry. Our repressor binds to the operator and blocks RNA polymerase from binding to the promoter. And so if the repressor is bound to the operator, then RNA polymerase can't transcribe the structural genes. So the operator is the region of the DNA where the repressor can bind to block the promoter. One last piece, the regulator is the piece of DNA that encodes for the repressor. So the repressor is another protein that gets expressed by the gene, which is the regulator. So let's write this out, and I think it'll be a little more clear. So if we have a stretch of DNA, we can have a region here called the regulator. This is our DNA. And this is going to make a protein called the repressor. So So the repressor can combine to the operator, and if it does that, it's found right here. Then RNA polymerase another protein, this is an enzyme. If our repressor is bound to our operator, RNA polymerase cannot bind to the promoter, and if that happens, then we don't transcribe our structural genes, and these do not get expressed. So we have our regulator, that's the stretch of DNA that encodes for the repressor, which is a protein. That repressor can bind to the operator, and if it binds to the operator, it blocks the RNA polymerase from binding to the promoter, and it blocks the expression of these structural genes. If this repressor isn't made, or if it's inactivated, which we'll talk about later, it won't bind to the operator, and then the RNA polymerase can bind to the promoter, and we can express our structural genes.
So is that kind of like a way of sectioning off which genes it wants to express?
Exactly. This is how we're going to regulate the gene expression. So we're only going to express these structural genes when we really need to, and so the rest of the class we're going to talk about why would a cell need to express certain genes at certain times and exactly how it would do that.
Does the regulator need to be turned into RNA and then sent out of the nucleus?
Yeah. Great question. So in between going from DNA to protein, we have the creation of RNA, and it goes to a ribosome and makes the protein.
Now, would the regulators need to be repressed themselves so that not too many repressors are being made?
We'll get to that, but that's it's like basically like the same process, but another layer up. So if this is a little confusing, just bear with me for the rest of the class. That's going to become more clear as we talk about two more specific examples of this kind of system. Any questions about the terminology before we go on though? All right. So we're going to talk about two types of operons, since we're going to go into a little more detail on what an operon is. So the first one we talk about is the lac operon. So in this case, bacteria is sensing its environment. It's going to figure out, all right, is there glucose there? If so, we're just going to use that and everything's fine. If there's no glucose there, but there is lactose, we're going to have to break down lactose to make glucose, and then we can use that. If there's no glucose and no lactose, this is not the right solution. We don't need this enzyme. There's another pathway that we'll go through. But essentially, the lac operon is going to be breaking down lactose when there's no glucose and not when there is glucose. Okay. So do you think if there is glucose present, it would be a good use of energy to make this enzyme?
No.
Exactly. It wouldn't be necessary. And bacteria has to really conserve its resources because it's always competing against other bacteria, so it has to do every little thing it can to make it more likely to survive. Lactose is a disaccharide, so this is two carbohydrates that are binding together, and they're composed of a glucose monomer and a galactose monomer. And so if there's no glucose present, what the bacterial cell can do is it can use this enzyme to break down the lactose to make glucose and galactose, and they can take the glucose and go do cellular respiration and everything is fine. If there's glucose already present, it doesn't need to do this. It would kind of be a huge waste of time and energy. So it only undergoes this process when there's no glucose present, but there is lactose. So what the cell really has to be doing is sensing its environment and determining, is this the right time to make this enzyme? If so, let's do it. If not, let's not do it.
Is that aerobic or anaerobic?
Well, this is anaerobic. Yeah, but breaking down the glucose to make ATP would be aerobic. All right, so this is called an inducible operon. So we're going to talk about inducible and repressible operons. The lac operon is called an inducible operon because it can be turned on, but by default it's off. So usually there's glucose floating around for whatever reason, and it doesn't need to turn this on, but it does turn it on when it's necessary. So an inducible operon is one that can be turned on, but it's off by default. So if it does transcribe these lactose metabolizing enzymes, which are the structural genes that we talked about over here, then it will create the appropriate enzymes to break down lactose. If it doesn't transcribe these genes, then it won't create those enzymes and it won't be breaking down lactose. So again, it's going to try to optimize its use of resources according to its environment. So let's talk about some of the mechanics of exactly how this is happening. So we have our regulatory gene, or our regulator over here. And so this is going to be making our repressor that we see up here. So like Matthew pointed out, there's mRNA that's made in the process that I didn't really draw up here. So this is going to produce what's called an active repressor. So this is a protein that got expressed from the regulator or the regulatory gene, and it's active. So it can go bind to the operator, and if it binds to the operator, it's going to prevent these structural genes from being produced. So when we look at the promoter and the operator region, we can see how it's interacting with lactose. So if there's no lactose present, we have the blockage of these structural genes. If there is lactose present, then we're going to want to be making these structural genes to be able to break down that lactose. And so the lactose is going to bind to our repressor and inactivate it. If it's inactivated, it can't bind to the operator. If it can't bind to the operator, then that means RNA polymerase can bind to the promoter, and then we can go ahead and transcribe this DNA and make the enzymes that are going to break down lactose. So we can see here that lactose is actually involved in the regulation process. So lactose can inactivate this repressor and allow the transcription of these genes to produce enzymes that then break down lactose. So this is like a feedback mechanism that makes more of these enzymes if lactose is present, and it doesn't make these enzymes if lactose is absent, like we have up here. Because if lactose is absent, then this repressor is active, and it binds to the operator, and it blocks transcription.
How heavily is this going around the final genome? Like in what depth?
You can look at the review sheet I posted today. What does this seem? Well, we have 2 days to do it. What seems confusing about it?
The terms, like, that's the confusing part.
We can practice those. Okay.
Although the words all sound the same.
Yeah. Does the concept of what the repressor does make sense? How it responds to the presence of lactose? So if the repressor is active, it blocks transcription. If it's inactive, it allows transcription, and it becomes inactivated by the presence of lactose. It's like the key takeaway from this. Does that help?
Again,
Okay.
It doesn't do anything, it's blocking transcription.
Yeah, so if the repressor is inactivated, it doesn't block transcription, and it allows the creation of the enzymes that can break down lactose. If there is no lactose present and the repressor is active, then it can bind to the operator and block transcription. So if we look at this little flowchart, we can kind of see if there's lactose present, the repressor is inactivated, the enzymes get produced, and lactose can be broken down into glucose, and then that glucose can be used to help the cell grow and do all these cool things that it likes to do. So the end result is that we only are going to produce this enzyme when there's no glucose and lactose is available. And so the cell kind of responds to those environmental cues and acts accordingly. Do we want to go over this a little bit more before we go on? Any questions?
So that other slide, it was wrong then, I guess?
No, why?
No, where you had said it binds to DNA to prevent transcription. It's just regularly?
What's that?
The other side where you said that it.
The TRP operon, or the trip operon, if you want to call it that, this controls the synthesis of tryptophan, which is an amino acid. So often tryptophan is found in the environment, and the bacterial cells can just take that up and don't need to make their own. But sometimes it's not found in the environment, and that means that the bacterial cell needs to start making its own tryptophan to survive. So the cells want to make tryptophan when it's not around, but when it is around, they're just going to take it from the environment and not make it themselves, and that's going to allow them to conserve their energy. So if tryptophan is in the environment, do you think we're going to be synthesizing enzymes to make more tryptophan? No. So now we can kind of see this process unfolding where we're going to have tryptophan coming back and acting on the transcription process to either make it more or less likely to occur. So this is the kind of the whole point of the chapter, is to see that even though all of our cells have the same DNA, they can respond to their environment and act accordingly. So we're only looking at the presence of lactose and tryptophan as kind of like a really simplified model of this, but cells also respond to different chemical cues like hormones and different types of signaling between neighboring cells and cells that are far away. And this is part of the reason that cells can perform very different functions in different parts of the body, and that organisms can develop from just one cell as like a zygote and grow into a really complex multicellular organism, because they can respond to different cues, because they'll be in different environments, and as they respond to different cues, they perform different functions, and now you can get all sorts of complex behavior because you have all these cells interacting with each other and doing all these different things.
Do cancer cells do any of this, or they just grow and that's all they do?
That's a great question. So what most of your cells do is they are really sensitive to these chemical signals, and they only grow and act when they're kind of told to. What cancer cells do is they just say kind of like, screw that, we're going to grow as much as we want, and they just express their genes whenever they want and not in relation to their environment. So cancer cells kind of get like decoupled from all of this chemical signaling in some sense, and then just do their own thing and grow whenever they want.
So chemical changes in the body have no effect on cancer. The only thing that has effect on cancer is like apoptosis.
Well, so I shouldn't have said, they still do respond to chemical cues, but kind of not in the same controlled way that your normal cells do. Yeah, so that's one thing that, one treatment option is that you send in chemicals basically that will interact with the cancer cells and either make them grow slower or undergo apoptosis. And there's actually a way to have chemical cues that only cancer cells will respond to and your normal cells won't, that help you kind of target those cells specifically. So this operon, this model, is a little bit different from the previous one that we talked about. So again, we're going to be making these enzymes only when tryptophan is not in the environment. So tryptophan it's an essential amino acid, so it's an amino acid that bacterial cells really need to survive. And if it is in the environment, they don't need to synthesize it themselves. If it's not there, they do need to start making it on their own. And so this is something that is on by default, but can be turned off when tryptophan is present. So is this going to be inducible, do you think? So we had inducible, off by default, can be on. So what we're going to talk about next is repressible operons. On by default. Can be turned off. So this is the opposite of the inducible operon. So when tryptophan is present, it's going to bind to our operon and activate it. And that's going to prevent the expression of these genes. So when tryptophan is there, we don't want to be making more tryptophan because that would be a waste of energy. So we don't need all of these enzymes. So we're going to allow tryptophan to activate our repressor and block transcription. When tryptophan is not there, the repressor is inactive, and so it's not going to bind to our operator, and we are going to make all these enzymes, and now we can synthesize more tryptophan, or synthesize tryptophan in the first place. So now we have a system where the cell can look in its environment, it can say, all right, is there tryptophan here? If not, I'm going to make some, and it's going to make some because it has an inactive repressor, and this inactive repressor is not going to block the expression of these genes, and then we're going to make more tryptophan. If there is tryptophan present, it's going to bind to our repressor and activate it, and we're not going to make all of these genes.
When the bacteria is looking at its environment, is it looking outside or inside or both?
So all it's really doing is it's letting some of the chemicals that are in its environment go into the nucleus. And based on the composition of the chemicals in the nucleus, we're going to have different rates of transcription occurring.
So it would make these until it has enough of these, and then it would turn it off.
Exactly. Exactly. Great. So if there is no tryptophan present, it's going to make all these enzymes, and it's going to start making a ton of tryptophan. And then after it's made enough tryptophan, that tryptophan will come back in the nucleus, and it will activate the repressor, and it's going to stop expressing these genes. So this is how the cell makes only as much tryptophan as it needs to undergo the vital functions. So the important things to realize are it's not really like all the individual steps. It's kind of like the overall concept is that cells regulate their gene expression based on the chemical environment around the cell in order to conserve resources. And it does it by changing the transcription rates of different sections of the DNA or our structural genes. And these structural genes, when they're expressed, are going to change their environment, and when they're not expressed, won't be changing their environment.
So in this scenario with the trip thingy, is it possible for there to be some of that in the environment, but like not enough for the bacteria, so it needs to make some as well?
Yeah. So remember when we talked about like enzyme speeds, and we talked about substrate concentration, and we had some sort of like plot like this where you have like a saturation point, you have like substrate, no, that's the wrong term. Substrate, concentration, and we have like enzyme speed. This is a really similar curve that we have here. If there's a little bit of tryptophan present, we're going to have some blockage, but not a ton of blockage. If there's a lot of tryptophan present to like a ton of tryptophan present, we're going to have like total blockage of transcription. So it gives the cell like some flexibility and precision with the control of this.
It says that when the repressor is activated, when tryptophan is present, it blocks it from synthesizing tryptophan.
Yeah.
So is that kind of like its way of mediation?
Yeah.
Mediating itself?
Yeah, so when there's tryptophan present, it's not going to make more because it has an active repressor that blocks transcription. So this is how all of our cells can have the exact same DNA but do all these different tasks because they can respond to their external environment. That's the overarching message. And then we have these inducible and repressible operons, two different ways of controlling them, but they still depend on whether or not this repressor is going to be activated or inactivated. And so I have some review terms for us to go over as you study them, and I have another table that goes over what happens if tryptophan is available versus unavailable, and what happens to the repressor and the promoter. And so tomorrow what I wanted to do is go a little more in depth in like a review worksheet that I made on what's involved in these tables, and I thought that would help us understand a little bit more what's going on. And also we can